Bismillahirrahmanirrahim. Assalamu alaikum dear all students. I am Zakirullah, lecturer in medical laboratory technology. Today we will learn about the quantification of glycated or glycosylated hemoglobin. This topic is in the subject chemical pathology. The main outlines of today's topic is the sample requirement for the glycated or glycosylated hemoglobin and the quantification methods and their explanation, the different methods which are used to quantify to measure the quantity of glycated hemoglobin. The specimen requirement for glycated hemoglobin, we need a whole blood sample that is well mixed with the anticoagulant, usually EDTA, ethylene diamine tetraacetic acid. This is the anticoagulant which is usually a calcium chelator, and this binds with the calcium, so the process of the clotting it is stopped, and the blood it cannot be clotted because we need red blood cells and normal red blood cells for this process. So in the whole blood, the red blood cells, their structure is maintained, and we can prepare a hemolysate from these red blood cells and then find out the HbA1c level in the red blood cells. The quantification methods of glycated hemoglobin, they are grouped into two major categories. The first one is based on the different charges between the glycated or the glycosylated and the non-glycosylated hemoglobin. First of them is the ion exchange chromatography. Second one is electrophoresis, and the third one is the isoelectric focusing. The second method is based on the structural characteristics of glycogroups on the hemoglobin. The first one of them is the affinity chromatography, and the second one is immunoassay. Affinity chromatography method. This is the method which is preferred in clinical laboratory for measurement of glycated hemoglobin. In this method, the glycosylated hemoglobin, they attach to the boronate group of the region, and from this region, this boronate containing glycosylated hemoglobin is selectively eluted from the region bed using a buffer. This affinity chromatography, this is temperature. This method is temperature independent and usually not affected by the other types of hemoglobin, that is hemoglobin F, hemoglobin S or hemoglobin C. This is the reaction of affinity chromatography method, in which a boronate affinity matrix bonded with the glycated protein of the hemoglobin, and they form a boronate glycated protein complex. This complex is eluted with the help of buffer from the region bed, and its value is calculated. The second method is ion exchange chromatography. In this method, the negatively charged hemoglobins, they attach to the positively charged region bed, and that glycated or glycosylated hemoglobin is selectively eluted from the region bed with the help of a buffer, and this buffer have specific pH in which the glycohemoglobins are the most negatively charged and they eluted first from the column. The issue with this method is this is a highly temperature dependent method, and slightly variation in the temperature, it can cause the disturbance or the decrease or increase in the result. And also, if there is other type of hemoglobin disorders or hemoglobinopathies, so this ion exchange chromatography is disturbed by other types of hemoglobin disorders. For example, the presence of hemoglobin F yields a false increased level of the glycated hemoglobin, and also the hemoglobin S and hemoglobin C, it produces false decreased level of HbA1c. So, this is the issue with this type of glycated hemoglobin quantification method. The third type of method is the immunoassay method. In this type of quantification method, the concentration of HbA1c specifically and the concentration of total hemoglobin are measured, and their ratio is then reported as a percent hemoglobin A1c. In this type of quantification method, the glycated hemoglobin F is not measured. So, if there is hemoglobin F more than 10%, so the amount of HbA1c will be lower than the expected, and that will be falsely low, because a greater proportion of the glycated hemoglobin will be in the form of glycated hemoglobin F. So, there will be issue with this method. This is the reaction of immunoassay method, in which a well which having anti-hemoglobin antibody, the blood sample of whole blood is added to this well, and there is immunoreaction that the antibodies to the hemoglobin, they are attached. After attachment of this antibody with the hemoglobin, another anti-HbA1c antibodies which is attached with the enzyme level detection antibody, that is also added to this. So, this enzyme level HbA1c antibody, it is attached. So, the amount of color produced by the immune reaction, it is directly proportional to the concentration of HbA1c present. If the reaction color and the reaction gives strong color, concentrated color, this means that there is a higher amount of HbA1c present in the blood, and if the color of the reaction is light or it is very low, so this means that amount of HbA1c that is low. So, this is the chemical reaction of immunoassay method. The last one is HPLC, the high performance liquid chromatography. and also electrophoresis. These methods are also used for the quantification of hemoglobin A1c or glycated hemoglobin. In HPLC, all forms of glycated hemoglobin, that is A1A, A1B and A1C, all types of hemoglobin, they are separated. This is the summary of the different methods for the quantification of glycated hemoglobin. The methods based on structural differences. First one is immunoassay method, in which a polyclonal or monoclonal toward the glycated N-terminal group of the beta chain of the hemoglobin, they are present, and these antibodies react with these glycated group. So the color of the reaction gives us the concentration of the HbA1c. In the affinity chromatography, the glycated and non-glycated HbA1c are separated, and this one method is the preferred method because this is not temperature dependent and this is not affected by the other type of hemoglobins. The three methods are the other four methods based on the charge differences. The ion exchange chromatography, in which a positive charge region bed is present and the glycated hemoglobins, they attached to this positive charge region bed. The issue with this method that this is a highly temperature dependent method, and also the other issue is this is affected by the different hemoglobin disorders. Electrophoresis is another method in which separation is based on differences in the charges. It is also affected by the hemoglobin F if its value increases more than 7%, so the results are disturbed. In the isoelectric focusing, the type of electrophoresis in which isoelectric point is used to separate multiple type of hemoglobin based on their charges. In this method, the pre-HbA1c interferes, and the HPLC method, it is also a form of ion exchange chromatography, and it separates the different types of glycosylated hemoglobin A1B, A1A and A1C. So these are the all methods for the quantification of glycated hemoglobin. The issue with these current methods is that there is no standardization. It is a continuous issue with all the methods, that there is no standardized method for the measurement of glycosylated hemoglobin, and there was no consensus on the reference method. No single standard is available to be used in the assay, and because of this issue, the HbA1c values, it varies with the method and also it varies with the labs. If a person having HbA1c with one method, it will give you one result, and with the other method it will give you the other result. So that's why it varies between the labs. The International Federation of Clinical Chemistry and Laboratory Medicine, IFCC, this federation have developed a common definition for HbA1c and a reference method, and in this method specifically it measures the concentration of the glycated N-terminal residue of the beta chain of the hemoglobin with the help of either high performance liquid chromatography and electrospray, mass spectrometry HPLC ESIMS, or HPLC capillary electrophoresis. These methods are developed by the IFCC for the standardization and measurement of glycated hemoglobin, which measures the N-terminal residue of the beta chain of the hemoglobin. These methods are only used to standardize HbA1c assays, but they cannot be used for the clinical measurement of HbA1c because of the high cost, because of the special requirement, because of the trained personnel requirement, and because of the high cost. So that's why this method is only used for the standardization of the HbA1c assays. That was all for today's lectures, and our today's lectures were prepared from the Manual of Laboratory Medicine AFIP and Clinical Chemistry Techniques Principal Correlation 6th edition by Michael Bishop, Edward Foddy and Larry Schaff. Thank you for watching the video. Hope you have learned about the glycated hemoglobin, its different values, the different methods and the issues, and the standardized procedure for the quantification of glycated hemoglobin. If you have any question, you can ask. Thank you. Assalamu alaikum.